Welcome back to Chapter 4. We're going to be talking about prokaryotic cell diversity. Chapter 5 will deal with eukaryotic cells, but we'll spend more time this semester with prokaryotic cells because those are the ones that we deal with in lab, and they are primarily the ones that will be the causative agents of the diseases we will talk about later in the semester. We talked a little bit about diversity in terms of cellular morphology, the shapes of the individual cells, and from the coccus to the bacillus, the vibrio, the spirillum, the spirochetes. And so we're going to look at a little more of different types of diversity in this chapter. So this is a bacterium. We have the size bar below it. Those are always very important. 1 micrometer is the length of that bar, so that bacterium is probably about 3 micrometers long. We can see that it has either flagella or pili. They're not labeled. And the typical bacteria we have in our labs that you'll have images of slides are around 1 to 2 microns long. And a micron is 1/1,000,000th of a meter, so super tiny. This organism happens to live in the deep sea, where there's very little oxygen. We're used to thinking about living things as requiring oxygen to survive. We certainly do. But many bacteria can grow in the presence of oxygen, and when they do, they metabolize very similar to the way that we do. But lots of others can survive with or without oxygen. When we get into the chapters on metabolism, we will talk about both aerobic metabolism, which requires oxygen, and anaerobic metabolism, which means organisms that can grow when there is no oxygen present. These organisms attach to the sea floor with nanocables to sense oxygen and be able to use it, but they can survive in the absence of oxygen. Halophiles, shown here. Halo means salt, and anytime you see that suffix phile means a lover. So it's a salt lover. And there are some bacteria that can just tolerate salt. They're halophiles, but they just tolerate it, but they don't require it. And other microbes actually require salt. And this archaebacteria, Halobacterium salinarium. So halo tells you it's a salt bacterium. Salinarium, it likes saline or salt. Nitrogen-fixing bacteria are critical to life on Earth. Here they're shown in root nodules. And to fix nitrogen means to bind that nitrogen gas. That is actually the primary gas in our atmosphere. Oxygen is present and we need it, but N2 nitrogen gas is the most abundant gas in our atmosphere. However, we can't really use it in that format. So these nitrogen-fixing bacteria can take in that nitrogen gas and reduce it by adding hydrogens and make it ammonia, NH3. And then NH3 can be incorporated into molecules like nucleic acid and protein. Rickettsias need special staining so that we can actually see them, and they are obligate intracellular pathogens. Obligate means must. It's inside a cell. Pathogens can cause disease. And so all rickettsias have to find a cell to get into in order to survive and reproduce. And so we have tick cells. Those are eukaryotic. The really big cell, that's the nucleus of the tick cell. And all these little magenta type dots, those are the Rickettsia rickettsii. In this case, it's an organism that causes Rocky Mountain spotted fever, and it is a disease that is transmitted through ticks. So the tick bites an infected organism, it picks up the bacteria, the Rickettsia, and the next time it bites another organism can transmit that illness. Chlamydia are also obligate intracellular pathogens. We see here an epithelial cell. The elementary body of the microbe gets in. They get encoded by the plasma membrane, form an inclusion. They start to replicate inside that host cell, and then they are released. So chlamydia trachomatis is a microbe that causes sexually transmitted illness, chlamydia. And so that is one that it would be the reproductive cell, epithelial cells that are infected by that organism. And the cell dies when the chlamydia uses all of the host cell ATP, which is the energy molecule we'll discuss in our metabolism chapters. So this can be very damaging to reproductive cells. Males typically have symptoms and get treated early. Persons with uterus and uterine tubes tend to have very little or no symptoms until the disease is very severe, and this bursting of the cell is what can cause sterility in people. Neisseria meningitidis, they are really fond of picking out the microbes that cause sexually transmitted illnesses. Neisseria meningitidis, in this case, however, causes meningitis. It is related to the Neisseria gonorrhea. They would look exactly the same on a chocolate agar. And it's called chocolate agar because it uses blood that has been boiled and denatured, and that causes it to turn brown. It doesn't actually have chocolate in it. This is an x-ray of someone with tuberculosis. So they've got the bilateral, both sides, pulmonary, lungs, infiltrate. So they've got the walled off features happening with tuberculosis. Groovy aspect to some prokaryotes is bioluminescence. So being able to glow in the dark. And that is a chemical that is produced in the cells that fluoresces. And so on slide A, we've got it streaked on agar, and you turn out the light and it actually glows in the dark. And this organism, Alivibrio fisheri, colonizes on squid and other marine life. And what that does is the bacteria get a nice place to live on the surface of the organism, and the squid get the luminescence to help it to track prey. You can sometimes see this bioluminescence in the ocean. I've seen it when I've been on a ship at night. You can see it in the wake of the waves. Surfers going through the waves will disturb the bacteria, and that motion will cause them to fluoresce. That bioluminescence is not only seen in this microbe. There are other prokaryotes and some eukaryotes that do this. Fireflies or lightning bugs do this. Jellyfish produce a luminescent protein. But in this chapter, we're looking at an actual prokaryotic microbe that can produce bioluminescence. Legionella pneumophila, or pneumophila, depends on who you ask. That P-H-I-L, a liver, and pneumo is lung. So this microbe really loves the lungs. It causes Legionnaire's disease, and it's shown here with a air conditioning unit. This was discovered at a Legionnaire's convention. So older soldiers at a meeting, and the microbes were living in the warm waters of the air conditioning units, and the droplets being released by those air conditioning units infected the lungs of these individuals and caused a severe pneumonia. Salmonella typhi causes typhoid fever. So it's a gastrointestinal illness, and gastro means stomach, and intestinal is your intestines. You'll also see enteritis, E-N-T-E-R-I-T-I-S. Entero means intestines as well. So there are other species of salmonella Salmonella is the genus. In this case, typhi is the species causing typhoid fever. There are other types of salmonella that cause gastrointestinal illnesses but are different species. You may have heard of Typhoid Mary, was a domestic worker in the 1800s who was a carrier for Salmonella typhi. She herself did not come down with the disease, but was unfortunately transmitted it to members of households where she worked, and she ended up being quarantined because she wouldn't stop working in houses and causing illness, and some people even died from the typhoid fever. Myxobacteria are really crazy. They form fruiting bodies. Under particular conditions, the individual prokaryotic cells come together and make these conglomerates called myxospores. And so it's a way for them to hang out together under unfavorable conditions, and when conditions are favorable again, those myxospores are released and then they can reproduce again. The spore is similar to what we saw with endospores in a previous lecture. And these are called myxospores because it's not that hard covered individual endospore like we saw with the bacillus cells. It is actually a conglomeration of organisms that are just hanging out in this protective sort of slime until conditions are favorable. Helicobacter pylori, called pylori for the pyloric valve in the stomach, causes gastritis, so it does not affect the intestines. It is affecting the stomach. They do have flagella. They are at one end, so they have a grouping of flagella at one end for moving, and they cause ulcers and stomach cancer. It was long thought that ulcers were caused by stress, and the stress and the corticosteroid release was damaging the stomach lining, and that might make it worse, but they actually isolated these bacteria from the stomachs of people with ulcers. The person initially doing this work actually ingested a pure culture of Helicobacter pylori, did come down with an ulcer, and they isolated those bacteria back out of him. It's truly not recommended science, but no one would listen to him or realize that his work was accurate. So that itis suffix means inflammation of. So gastritis is inflammation of the stomach. Enteritis would be inflammation of the intestines. Encephalitis would be inflammation of the brain. So Helicobacter pylori does cause ulcers. Spirochetes we saw in our microbial morphology. So they actually have, in this scanning electron micrograph, in the cartoon we can see it has this filament that gives them this corkscrew appearance, and they do spin as they move through liquid media. And when you cross-section them, you can see these little endoflagella. So they have flagella, but they are not attached to the outside of the cell. They are within the cell membrane, and they have that nucleoid, so they don't have a nucleus. There is no membrane surrounding that. That is just where their DNA is sitting, and they've got the cytoplasm. The fact that we have a cell membrane and an outer membrane, anytime you see an outer membrane on a prokaryote, means that it is gram-negative. If you go back to our chapter on the cell wall structure, the gram-positives have their cell membrane and a very thick cell wall. The gram-negatives have their cell membrane, a thin cell wall, and then a thicker outer membrane. So the Treponema pallidum is a spirochete. It is a cause of syphilis, and it is one that is extremely difficult to culture on agar. Even if we were in a face-to-face lab, we wouldn't be working with spirochetes, not just because they are a causative agent of syphilis, but because they are extremely difficult to grow. on agar. You need very special techniques to culture them. Bacteroides, oid, kind of like, they are rod-shaped, so their morphology is bacillus. These particular bacteroides are from the human gut. They make up about 30% of the normal microbiota, so small living things. So our genome is our DNA. Our proteome is the entire library of all the proteins in our cells. And our microbiome are all of the microbes that live on or in us. So our skin is covered in microbes. Our intestinal tract is covered in microbes. The upper part of our respiratory tract, the upper part of our digestive tract, our mouths. Someone had posted in the discussion that they were just kind of amazed. Are microbes really everywhere? Our ubiquity of microorganisms lab? And the answer is yeah, they are everywhere. We do not live in an abiotic or aseptic environment. Our skin is pretty well coated with microbes, and mostly Staphylococcus epidermidis, but also other microbes. There are also some eukaryotes. There are some fungi that are just a normal part of living on our skin. Our mouths have a lot of streptococci, similar to the organisms that cause strep throat. They have the same cellular morphology and arrangement, but they lack features that would allow them to cause disease under normal circumstances. We have bacteroides that live under our gums. We have lots of different microbes. And by numbers, we have more prokaryotic cells living on us than we actually have human cells. Now, of course, by weight, we have far more human cells. But by numbers, we are teeming with prokaryotic cells. Planktomycetes. What a crazy name. Usually when you see M-Y-C-E-T, it means a fungus. And plankto, like plankton. These are found in environments. To be sessile means that they don't really move around. So instead of this being a flagella, it's a holdfast. It's a clumping of appendages that allow them to stick to surfaces in that aquatic environment where they live. The ones in a different life stage of this cell don't have the holdfast, and they can use that appendage for swimming. Now, 500 nanometers is the size bar on this one. 1 nanometer is 1/2 of, well, millionth. Nope, 1,000th of a micron. So if you have 1,000 nanometers, you have 1 micrometer. So this is half of a micrometer here. So it's not quite a whole micrometer long. Super tiny. Here the space bar is 100 nanometers. Why a different space? I don't know, but let's see, one. It's also probably around 600 nanometers across. This really gunky looking thing is called a Winogradsky column. And you can check out the subtitles, the captions for this. I'll make sure it is spelled correctly. Winogradsky column, because Dr. Winogradsky figured it out. Essentially, you gather the mud from a pond and some pond water in a tall glass cylinder, fill it to the top, a little bit of airspace at the top, and then you seal it off so that no more oxygen can get in, and then you just let it sit and do its thing. So there will be nutrients in that water and in the mud so that the microbes can use that for energy. We have here, they're showing the purple and green sulfur bacteria. They use chemicals called bacteriochlorophylls to do photosynthesis. They are prokaryotic, so they don't have chloroplasts, but they have inclusions that concentrate these pigments. The green bacteria have green chlorophylls. The purple bacteria have purple ones. And we put this in a window so that the sunlight allows these microbes to do photosynthesis and convert that sunlight energy into chemical energy that will feed the other microbes that are in here that cannot photosynthesize and need that products from the photosynthesis. And a Winogradsky column can be self-sustaining for years and years and years because the photosynthetic microbes keep harvesting sunlight, producing the chemicals that the non-photosynthesizers need, and then the waste products of the non-photosynthesizers, who metabolize a lot like we do, produce carbon dioxide and water that the photosynthesizers need as their carbon sources. On the right, now we have 20 microns as our space bar, so this is an extremely large prokaryote. It's Microcystis aeruginosa. It's a form of cyanobacteria. These were formerly known as blue-green algae. Blue-green because of the pigment color, and algae because they were so big. It was thought that all of these green dots were chloroplasts and were just obscuring the nucleus and we couldn't see it. But genetic testing indicated that these were indeed prokaryotes. They have all of the features of prokaryotes. Their DNA is organized like prokaryotes. They don't have chloroplasts. These are just inclusions. And in warm temperatures, cyanobacteria can multiply and produce neurotoxins. Those algal blooms, we see that definitely in Humboldt County, particularly around still water like ponds, lakes, lagoons. There'll be the toxic algae warnings. Swimming in it, not terrible. You wouldn't want to get the water in your mouth. That's why it's really recommended that children and pets are kept away from the waters when there could be a cyanobacterial bloom, because they won't be as careful, get water in their mouth, and it would not take as much of those neurotoxins to cause severe illness or even death. So we have an electron micrograph, Actinomyces israelii, because discovered in Israel. Again, that MYCE, usually used to describe fungi. This happens to be a prokaryote, fungi are eukaryotes, was called Actinomyces because it has this branched structure that looks a lot like the way fungi reproduce. But again, genetic testing proved that it belonged to the domain of bacteria. In B, we have Corynebacterium diphtheria. I love it when the name of the microbe tells you the disease that it causes. And so palisades are when they make these little V shapes. So again, we don't have any Corynebacterium that we would work with even in a face-to-face class. Number one, we don't want diphtheria. There are different species of Corynebacterium that are not disease-causing, but they are again super difficult to grow and they're difficult to stain. And in C, we have Gardnerella vaginalis. The large cells are epithelial cells from the vaginal wall, and the dots are the Gardnerella that surround it. So this was from a pap smear. The person that they sampled this from had vaginosis. And so itis means inflammation of. Osis means a condition of. So a vaginal condition. Pap smears are called that because they are done to look for cervical cancer. And cervical cancer is often caused by human papillomaviruses. But they can also discover things like a bacterial infection in that person. And so this Gardnerella vaginalis can just be an imbalance in the normal flora of the person, the normal microbiota. But it does cause, when they're overgrown, severe itching and symptoms that are similar to a vaginal yeast infection. But it's important to have that diagnosis because the treatment for a yeast infection would be very different for the treatment for a Gardnerella infection, because Gardnerella is prokaryotic, it's a bacterium, and yeast are eukaryotic, they're fungi. So drugs that combat fungi usually are ineffective against prokaryotes, and drugs that combat prokaryotic infections are generally ineffective against eukaryotic infections. Clostridium difficile, or difficile. Difficile means difficult, and you may have heard of this, those of you who might already be working in the healthcare field. C. diff. C for the abbreviation for Clostridium, and diff, shortened difficile. It is gram-positive, meaning it has a very thick cell wall. It's rod-shaped, so it's a bacillus. Causes severe colitis, so an inflammation in the colon. And it often happens because the normal gut microbiota has been destroyed by antibiotics. So overuse of antibiotics could certainly cause this. There are conditions that people may suffer from where continued long-term use of antibiotics might be necessary to save their life. And in the course of that, the antibiotics are indiscriminate. If it's killing one gram-positive organism, it might kill others. It might wipe out the gram-negatives. And if the person had some of these in their normal microbiota, most of us do, if we eradicate all the others living in that colon, then these overgrow. And it does cause severe colitis, bleeding, ulcers. It can be life-threatening. And the most recent treatment, because more antibiotics are not really a good idea, is a fecal transplant. To take fecal matter from someone with a healthy intestinal flora and put that into the colon of the person suffering from C. diff. And then the bacteria contained in the healthy feces will help sort of recolonize the colon in the person suffering from Clostridium. So this is a gram stain. I know that you're all already frustrated at not being able to put your hands on the materials in lab and do the experiment yourself. Watching somebody else do it really is not the same. And believe me, I empathize. I tried so hard to get you guys kits, but all of the manufacturers cut us off in May and just said, we're not doing any new kits. Bummer. So the bad news, you don't get to play with stuff yourself. The good news is gram stains are really frustrating to do, and we're going to have some very nice pictures of them. So gram stains do detect that cell wall thickness. The gram positives are called that because they positively hold on to that first stain because they're very thick cell walls. The dye gets trapped in there and can't be washed out of the cytoplasm. This happens to be Streptococcus pyogenes. So coccus, the individual cells are round, and strepto, the arrangement of the cells, is in chains. Pyogenes means it generates, in PO, fire, generates fever. It's a fever-causing streptococcus. It's also the organism that causes strep throat. So that is a stain through the microscope. This is an image of the bacteria that have been streaked across the surface of blood agar, and they lyse the red blood cells, causing this clearing of the blood around it. And so you will get to see other organisms streaked onto blood agar later in the semester and see this type of pattern of the lysis of red blood cells. So we have Bacillus cereus. We've got violet rod-shaped cells are the B. cereus. Those are the big purple ones. And then we have small pink ones are gram-negative. So this is an intentionally mixed culture. If you were doing a gram stain on a culture that you thought was pure, meaning containing only one species, and it looked like this, you'd be pretty upset There's very obviously two different things in this culture. But if we look at Bacillus cereus and streak it on blood agar, it makes these really sort of crusty, dry, irregular-edged colonies. This is a scanning electromicrograph of Staphylococcus aureus. You may have heard of Staphylococcus aureus. It is the causative agent of MRSA. That's methicillin-resistant Staphylococcus aureus. There's also MDRSA, which is multi-drug resistant Staphylococcus aureus. So again, the coccus part of the genus tells us that the individual cells are coccus, or round, and staphylo is that prefix that means in clusters. So they're called grape-like clustering. We won't see too many electron micrographs in our pictures from lab. We will see regular light micrographs, but we will still see clumps of round circles. So staphylo means a cluster. Coccus means those individual cells are circles. This is M. tuberculosis. Again, the name tells you what it causes. The M is for mycobacterium, and the myco in this case stands for mycolic acid, which is very waxy. So these are gram-positive organisms, so they have a thick cell wall made out of that peptidoglycan, and outside of that, or within their cell walls, they also contain mycolic acid, super waxy. And again, in a face-to-face class, we obviously would not culture Mycobacterium tuberculosis. It's definitely above my pay grade to do that. But the other reason why we wouldn't culture other mycobacterium that do not cause serious disease is that wax makes them grow super slowly. If we were to take most bacteria, take a little dip and streak them on agar or put them into broth, within 24 hours we would have billions and billions of microbes. If we were to streak Mycobacterium tuberculosis or other species of Mycobacterium onto agar, it takes 12 to 24 hours for them to double in size. So if we put 100 bacteria on a plate, by morning we'd have 200, which would still be invisible. So they are super slow growing, which gives them some advantages. Most drugs work on rapidly growing microbes, so they don't work against slow growers. They also grow slowly, so it takes they can be spreading to various organs or throughout the lungs before the body can recognize that there's a lot of microbes present. We once again get back to our phylogenetic tree of life. There it is, our domains. So we have the bacteria as branching off here. The archaea, the ancient ones, are more closely related to the bacteria than they are to the eukarya, but the eukarya are more closely related to the archaea than they are to the bacteria. And again, those classifications were based on ribosomal RNA sequences. So by looking at the ribosomal RNA, the RNA molecules that are in the ribosomes, those little sandwiches that will read RNA to make proteins, the genetic sequence puts all living things into three categories. So we are here, meaning we are most closely related to fungi and plants, maybe some ciliates, and the bacteria are way over here, and then the ancient ones in the middle. So Deinococcus, C-O-C-C-U-S, it is round. Radiodurans, so it's durable to radioactivity. Conan the bacterium survives the harshest conditions on Earth. Can't even irradiate it to death. It's the cockroach of bacteria. And then we have this example of Sulfolobus, which is able to oxidize sulfur. and it will store those sulfuric acid in its granules. So that is a pretty tough little microbe. It's also about a little over 1 micron big, and it does metabolism unlike any type that we are going to discuss in this class. If it's capable of doing this and storing sulfuric acid in its cells, it is not going to be a microbe that would find us very hospitable. So there aren't any organisms like this that would be able to cause disease in humans. An image of halobacteria, remember salt-loving, give them this distinct purple color. If you've ever looked down when you're flying into SFO, there are great ponds of halobacterium that you can see these colors in it. That wraps up our prokaryotes. So you've watched the video. Just like in the first week, your next step is to post some sort of thoughtful question or comment of about 100 to 200 words with a title. Do that by 11:59 a.m. and then return to respond to 2 of your classmates. Nearly everybody did that. A couple of people missed responding to that second person, but you'll get the hang of it, and we will have the exact same formatting throughout the semester so that you know what to expect from your lectures. So make sure you do your comments and make sure that you look for the videos, the pre-lab videos to watch for this week's labs.